Dr. Jenelle Dunkelberger, who is a geneticist at Tilfix Norseman, presenting for us. Dr. Dunkelberger holds a PhD from Iowa State University, where her research focused on the role of host genetic pigs in response to viral diseases. She continues to study this topic along with other health-related issues as head of the Tilfix Norseman Global Health and Behavior Research Platform. Today, Dr. Dunkelberger's presentation will be on breeding strategies for improved health and welfare in pig production. So I'll turn it over to you now.
All right, thank you very much for that nice introduction and the opportunity to speak with you today. So yeah, really appreciate the chance to kind of get to know your group a little bit and looking forward to the interaction. So if at any time you have questions, please feel free to stop me. I don't know if I can see hands very well from my view, so you may just interrupt me to let me know if I need to pause, because the camera looks like it's hovering above a little bit, so I can't see hands too well, but I'm sure you'll let me know. Yeah, so I'm going to give a little bit of an update here in terms of some of the breeding strategies that we are working on developing for health and welfare related traits at Topics Norseman. And in case Topics Norseman is new to you, if you had not heard of Topics Norseman before today, I thought I would just put a few background slides here to give a little bit of an introduction or background to who we are as a company. So we are a swine breeding company. We are a global genetics company. The headquarters are actually in the Netherlands. We have research headquarters in both Norway and the Netherlands, but the business headquarters is in the Netherlands. And that's because this is actually the product of a merger between two independent breeding companies, Topigs and Norseman. So Topigs coming from the Dutch side, from the Netherlands, and then Norseman coming from Norway. So these two breeding companies merged in 2014, and yeah, we've been operating under that name since. Then I also included a couple of figures here to give a little bit of maybe a flavor of the culture or kind of who we are as an organization that I think makes us unique. One is that we invest heavily in R&D around innovation. So we're really focused on trying to identify ways to improve data collection for existing traits within our index, but also looking at developing novel traits in ways of phenotyping. And we have a substantial research and development budget for that, which is really driven by the ownership structure of our company, because we're not publicly traded, but rather we're owned by Dutch and Norwegian pig farmers. And so part of their reward, I guess, in terms of operating this company is the benefit that they see from investing in improvement that is very tangible for them to be able to see in their own farms. This will look familiar to you, the genetic pyramid, but maybe just to emphasize that this is kind of, in a nutshell, how the business operates. Right, we have the purebred sire lines and maternal lines at the top of the pyramid. The parent female line, which for us is called the TN70, is an F1, so a hybrid of the landrace and also large white female. Then she's mated to one of either one of our purebred sire lines or a different sire line to create the three-way commercial crossbred pigs, which are the animals that end up going to market. This is an overview of our genetic portfolio. So we have a few different purebred maternal lines and a few different terminal lines, but in North America, we only market half of the products here. So you would only hear about the Z line or the Norseman landrace in Canada, the US, Mexico. The Z line being a large white female, and the Norseman landrace is a landrace from Norway. On the terminal side, we have the TN Tempo, which is a synthetic line. So yeah, we could also call it composite, mostly large white base. So that's our white boar. And then the TN Duroc, which is purebred Duroc. In terms of structuring our research group, we actually do that by dividing tasks and projects across 6 different research platforms. So each of these platforms has a lead and then a team of researchers working underneath that platform to address projects pertaining to that specific research area. And it's also kind of structured to make sure that we are focusing on improvement through the different parts of the pork production chain. So we have reproduction, health and behavior, nutrition, meat and climate or sustainability, genetics and genomics, and precision phenotyping. So I'm responsible for number 2 here, the health and behavior group. So I'll talk about some projects that we have addressing both the health segment and the behavior segment, but I'll start here with the health side. So our overall goal for the health segment is to breed pigs that exhibit intrinsic robustness to challenges. So that includes improving robustness under conventional conditions, but also under disease challenge conditions specifically. So starting with conventional health, one of the main issues in this particular category at this time is sow mortality or sow longevity, especially for the US industry. And this is a figure here showing the industry average sow mortality throughout the last decade. So you can see there's been quite a steady increase in the industry average mortality rate beginning there around 2014 and just kind of steadily increasing year over year. Recent results are now available from 2024, which show that that average has now surpassed 16% sow mortality. So this is a pretty critical issue, and you can see quite an area of opportunity for making improvement. Several major consequences, of course, of mortality. One is simply that it impacts sow farm productivity. So, in North America, but also globally, we have this challenge of finding and retaining qualified labor. So that in and of itself is already an issue. So anything that impacts the productivity or the day-to-day tasks on farm that could impact productivity are going to be a pain point. And so hauling out dead sows is a big disruption to the workflow when that extra labor just simply isn't there to deal with those sorts of interruptions. And of course, it's also hugely impactful on the bottom line of the producer. It's also inconsistent with sustainability goals, right? Because mortality is a welfare issue in and of itself. And it's also very much inconsistent with this need to produce more with less. So we need to have robust sows, meaning animals that will thrive in current and future production environments, leading to more sustainable pork production. But from a genetics company standpoint, actually breeding for sow longevity is not easy to do. And that's mostly related to the structure of how we house and rear our most genetically elite animals, which we do in a nucleus environment. So one of the aspects of our nucleus herds is that we have a high turnover rate. It's an artificially high turnover rate because we constantly want to be bringing in the most recent generation of genetics as a way of evaluating the most recent animals for which we're making genetic improvement. But as a consequence, we don't provide those animals with the opportunity to express their full genetic potential for a trait like longevity, right? Because they're removed before we're able to properly evaluate that. Another kind of necessity, but also challenge, is that we do everything that we possibly can to optimize the environment of the animals in these nucleus locations. Right? So we make sure that they have the best health status, the best management, the best feed, etc., all with the objective of having these animals or providing them with the ability to express their full genetic potential. But then as a consequence, we don't get the opportunity to evaluate their robustness to challenges. And so in both of these aspects, we don't have the opportunity to improve what we don't measure. So here's the situation at the nucleus farm level. We routinely collect tissue for genotyping from every animal at that location, which we use to get the genotype information. But as mentioned, we really don't have the opportunity to collect good robustness phenotypes at this level. However, at the commercial farm level, we have all kinds of opportunities to evaluate robustness, because that's where we see a number of challenges. So plenty of opportunities here to get the robustness data. So if we collect tissue from animals at this level, which we use for genotyping, we can link the information at these two farms based on their genetic relationships. So that allows us then to estimate breeding values for animals at both locations. And that's the approach that we use to estimate breeding values for traits like sow longevity. So we actually have two different longevity traits in our index, longevity to parity 2 and longevity to parity 5. You can see the heritability estimates here. So our structure is that we have several commercial farms in the US where we are collecting these robustness data, and then that's what we're using kind of as our reference population to estimate these breeding values. And this is showing a 5-year genetic trend for selection for longevity to parity 5. You can see that we can, although lowly heritable, we can make nice genetic improvement in this trait. This is a breakdown of different reasons associated with sow mortality and longevity, or sow mortality in the US. You might have seen something like this. I actually adapted this from a publication from Iowa State University, looking at classifying these different reasons for sow mortality. So the number one category here we see is unknown or other. Number 2 is lame, injured, or downer. And then number 3 is vaginal and uterine prolapse. So I think it's quite interesting here to realize that 83% of mortalities can be explained by these top 3 reasons. I've queried a number of different producers, big systems, smaller systems, and this feedback appears to be pretty consistent, that these are consistently the top 3 reasons for sow death loss in their systems, but then the order of these might change. So there's not a lot that we can do about trying to improve this number 1 category when we don't have more information about what the specific underlying causes are, apart from just simply selecting for better longevity in general. So that approach that I just described about collecting data from these reference populations and selecting for better longevity in general, that's how we're approaching this number 1 category. But now what I'm going to do is I'll talk about some other projects that we have in play looking at categories 2 and 3. So looking at number 2, related to feet and legs. So first of all, I think it's important to realize that structural traits are heritable. So we routinely estimate breeding values for over 30 different traits related to structural issues. Not all of those are included in our index, and of course, which ones are included actually depends on the line. And they range in heritability, as you can see, from being somewhere between lowly and moderately heritable to being quite moderately heritable. And the range that you see here is simply reflective of differences in heritability across different lines in our portfolio. I'm going to focus here on osteochondrosis in the next several slides because this is an important area of focus for us due to the fact that it's one of the main causes of leg weakness. In summary, this is an issue where it causes decreased blood supply in the growth cartilage. That results in all kinds of issues for the animal, ultimately resulting in a lot of pain and lameness. So we've been scoring for osteochondrosis since 1990, actually. And that was done initially by performing postmortems. So all of these phenotypes had to be generated by posting the animals and then doing a visual evaluation at 8 different joint locations. So very labor intensive, as you can imagine. And then in 2008, we launched use of the CT scanner in Norway. And several years later, then moved towards this approach of actually using images from the CT scanner as a way of evaluating and scoring OCD, rather than having to do these postmortem inspections. So now what we have in the index is actually an overall score or a summary score based on OCD scored at 8 different locations. So the lower the score, the better. And I think it's really interesting also to realize that, as you might imagine, there's actually a naturally antagonistic relationship between growth and osteochondrosis. And what we see is that animals that have fast early life growth, those animals are genetically predisposed to having an increased risk of developing OCD. So what I'm showing here is actually a series of genetic correlations between growth and OCD over the lifetime of the animal. So from birth till approaching slaughter weight. Sorry, this is in pounds, but that's what it's showing. And then growth here is expressed as days in phase. So the fewer the days in phase, the faster the growth of the animal. So fewer days in phase, faster growth, higher OCD. And you can see the magnitude of that relationship decreases as the animal gets closer and closer to slaughter weight. And that makes sense because most of the bone development is happening early in life. So this also means that in a breeding program, if you're putting a heavy emphasis on fast early life growth, you will increase susceptibility to OCD as a result if you're not explicitly directly selecting for reduced susceptibility to OCD. So we also have made the decision to explicitly select against OCD, as I mentioned, already beginning several decades ago. And you can see that we've got some nice genetic improvement in that trait. So next steps are actually identifying ways that we can automate this process, because even taking a CT image and deriving scores from those images is quite labor intensive too. So this next phase involves using machine learning to detect and then score those different joint locations. And then that also gives us the ability to actually extend that to including additional joints than the ones that are already included. So to summarize our approaches here, looking at improving feet and leg quality, we see that structural traits are heritable. Heavy emphasis on growth rate will negatively impact osteochondrosis if we don't explicitly account for that. and digital tools enable development of new and better data collection pipelines to facilitate genetic improvement in leg health. The number 3 category here that we are going to focus on is vaginal or uterine prolapse, which you may also hear summarized as pelvic organ prolapse or POP, as I'll refer to it. You've probably heard of this one too, because this has been, say, at the forefront of a lot of, say, industry news and communication over the past number of years, since it's an issue that's really seemed to increase in prevalence. There's a lot of suspected causes for this. I put a few of them here. So a number of different environmental or non-genetic factors, from mycotoxins to disease, water quality issues, etc. But of course, our interest was really trying to dig in and understand a potential genetic background for this issue. So to do that, we used data collected from purebred top-mixed Norseman females at a commercial multiplication farm in the US. And this is a farm where we already had a routine process for collecting tissue for genotyping and detailed phenotypic recording for removal reasons in place. So this already generated a nice data set that we could use to run this type of analysis. So we analyzed POP as a binary trait, so whether the sow was removed due to POP or removed due to another reason. Had just over 16,000 records available for the analysis, and in this particular data set, the incidence rate was 8.5%. This initial analysis was conducted just using pedigree data, and we saw that the heritability of POP was estimated at 22%. So to summarize results from this work, we saw that POP was a moderately heritable trait. So genetic selection can and should be used part of the multidisciplinary approach to reduce the incidence of POP. We also made a decision as an organization to implement direct selection against susceptibility to POP in January of 2021. So we've now been directly selecting against that for 4 years. But when I discuss this issue with producers and other stakeholders in the industry, I still also try to emphasize that genetic solutions, or this genetic approach, is certainly part of the solution, but this alone is not going to solve POP. Right, at a heritability estimate of 22%, we know that most of the phenotypic variation is still due to those environmental factors. So we got to continue to work on identifying and mitigating those potential irritators as well. Some remaining questions that we had from this work: would the heritability estimate of POP change if we use genomic rather than pedigree data? And we're also interested to investigate whether we could identify specific genes or genomic regions associated with susceptibility to POP. So for this part of the project, we had a collaboration actually with Iowa State University. So Jack Deckers and Jason Ross were involved, and Vishesh Bhatia, who was a PhD student of Jack at the time, worked for us as an intern for one summer. So he worked over in the Netherlands, actually analyzing the same data set that I just described. The only change being that he did a little bit of editing here in terms of the animals included, so reducing the overall sample size to just over 14,000 sows, to also make sure that we're restricting the animals in the data set to those with genotype information. So every animal was genotyped with either the 25K or 50K chip. If 25K, they were imputed up. So just as a reminder, results from that initial project showed that heritability was estimated at 22%. And then when Vishesh reanalyzed these data using genomic information, he observed that the heritability of POP was 35%. So the conclusion from this work being that POP appears to be even more heritable than what we had previously realized. So the next part was, again, looking at trying to identify if there are specific genes or genomic regions associated with susceptibility to POP. So he performed a GWAS for this part of the project and did identify some regions that we would consider to be statistically significant. And so here's his significance threshold. And actually also looked at tried to identify candidate genes based on predicted functional mutations. This is just a summary of these significant regions detected. So there were 6 of them across the genome. You can see here the chromosome and megabase location, as well as percent of genetic variants. So altogether, explained 9% of the total genetic variants. But as you can see, there really wasn't a single region that had a large effect on POP. There's more details available in this publication that Vishesh is the leader author on that came out last spring. So to summarize results from kind of part 2 of this POP work, results from this study confirmed that there's a genetic basis for susceptibility to POP. His analysis also showed that POP is a complex trait, right? So lots of genes with small effects, meaning that, of course, we can still make genetic improvement in POP, but it changes a bit our approach in terms of how we implement selection against POP. Can't use a single gene type of approach to cull animals with a certain undesirable genotype, but rather it requires that we continue to collect data on an ongoing basis to estimate breeding values for POP. So that's what we do. I mentioned that we implemented direct selection against POP in 2021, and we see that we've got a nice reduction in genetic susceptibility to POP since then. And actually, this trend was even already moving in the favorable direction even prior to implementation against that trait, probably as a correlated response to selection for our approach to enhancing cell longevity in general. So that kind of summarizes some of the work that we're focusing on currently, looking at improving health and robustness under conventional conditions. And now I'll highlight a little bit here our approach to enhancing robustness under disease challenge conditions specifically. So one of the reasons that we are really interested in improving robustness under disease is because of the direct relationship that this has with sustainability. So some of the main themes or goals of sustainability include reducing carbon footprint, reducing antibiotic usage, and raising happier and healthier animals. But in the presence of disease, that really compromises all of these goals. Because animals that are sick, they're not efficient. Those animals have poor feed efficiency, so they require more natural resources. Sick pigs also require antibiotic treatment, of course, and an animal that's not healthy and is diseased is certainly not experiencing its optimal health or happiness. So there are a couple of different approaches that we can take from a genetics perspective in terms of breeding for enhanced robustness to disease. Right, so one would be gene editing, and another approach, traditional breeding. So gene editing is a good tool when the goal is to introduce novel genetic variants. Right, and some different applications might include using that to perform gene discovery work, right, in a lab-based setting, or that can also be applied into the commercial breeding program. Traditional breeding, of course, is an alternative approach which makes use of existing variants within the commercial population. So this is the approach that we and every other major breeding company has already been using for the last decade to facilitate genetic improvement. Our approach to enhancing robustness to disease is that we're focused on enhancing robustness to a select number of specific diseases. That includes PRRS and strep suis. But actually, our larger goal is enhancing overall robustness to disease. So simply breeding for animals that are better at responding to whatever type of disease challenge they might encounter. Right? So better general immune response. And doing that using traditional breeding strategies. So here's some of the reasons that we're really focused on this concept of breeding for overall robustness, even more so than specific diseases. And that's because we want pigs to be able to thrive not only in their current production environment with all of the things that we know about, but also in future production environments, which could include pathogens that haven't even emerged yet. That's the only way that we can prepare animals for that type of scenario. Also, reducing dependency on antibiotics. So pigs that are naturally more robust to a variety of disease-causing agents, they're just going to be more, they're more robust and therefore have less dependency on antibiotics by definition. And then lastly, because we serve a global market, right? So the pathogen challenges or even the strains of a particular pathogen are going to vary depending on the region that the pig is produced. There's already a lot of evidence of naturally occurring genetic variation in response to disease. Now, here's just some examples of major diseases in pigs. But we've also investigated this from our own research trials. And one of the first ways that we started exploring this was actually by evaluating differences in robustness between genetic lines. So our objective was to look at differences in robustness between pigs that were sired by a Topics Norsvin sire line versus those from a competing genetic source. So here's just a summary of mortality from that particular study. So pigs that were sired by Topics Norsvin sire line versus a competitor line, looking at the cumulative mortality over time. So we experimentally inoculated each pig with a highly pathogenic strain of PRRS, and then they were co-infected with a number of other secondary bacterial infections. So this was a hefty challenge. As you can see, the ending mortality rate was 13% versus 22%, and that difference in mortality rate being highly statistically significant. So from this, not only the result for mortality, but also differences in finishing performance was a clear indication that, yeah, there are differences in natural robustness between lines. So then our follow-up question was, okay, if we detect this variation in robustness between genetic lines, what does the extent of that variation look like within a genetic line? Because that's really the more relevant question for us in terms of making genetic improvement, is seeing the extent of genetic variation within our own line to breed for enhanced natural robustness to disease. And substantial genetic variation even within a line is what we observed. So what I'm showing here is actually the percent progeny losses per sire for all of the different sires that were used to produce animals for one of our disease challenge projects. So all of these sires are of the same genetic line. And you can see that now some of these sires had very high percent progeny losses. Right? This one as much as 40% of its progeny died on test, versus some other sires that lost 3 to an even 0% of their progeny. When we estimate heritability for mortality under challenge, we saw that in this particular trial, the heritability of mortality under challenge was estimated at 10%. So this was kind of our initial look at trying to get some kind of a feel for the genetic variation in this trait. And after doing several rounds of these trials, we started to think about, are there ways that we can make updates or modifications to the experimental design to be actually able to identify whether there's even more genetic variation than what we've already detected. So one of the ways that we did that, or modifications to the protocol that we made, was to select sires that we use to produce the progeny based on our prediction of differences in robustness. So we used data from a couple of previous challenge trials as a training population in order to derive breeding values for robustness for all boars at a single boar stud. Then we ranked those boars based on their breeding values for this robustness trait. We identified the 30 lowest ranking boars, so with the lowest breeding values for robustness, to be the sires of what we called this low robustness progeny group, and then the 30 highest ranking boars to be sires of this high robustness group. You can see here we ended with approximately 650 progeny in each of those groups. So we took those two sets of kind of extreme progeny and put them through a similar challenge protocol as to what I already described. So those pigs entered the barn. We experimentally inoculated them with a highly infectious dose of PRRS. They were co-challenged with all their kinds of secondary pathogens, and we collected detailed individual phenotypes up until market. So here's a reminder, results from our initial trial indicated that heritability of mortality under challenge was estimated at 10%. And then when we made these modifications to the experimental design, we saw that the heritability actually increased to 26%. Right, so in this case, those modifications to the experimental design of really making an attempt to create these contrast groups as a way of creating even more genetic variation among the progeny was successful. You can also see that reflected here in terms of the mortality between these two groups. So this is showing the number of mortalities per progeny group from 0 to 3 weeks post-inoculation with PRRS. So this is the most critical time period in terms of morbidity and mortality post-infection. The low robustness group is shown here in the gray bars and the high robustness group in the pink bars. And we saw that twice as many low robust pigs died during this time frame compared to those in the high robust group. We also saw that pigs in the low robust group required significantly fewer treatments than those in the high robust group. So fewer deaths, but also fewer antibiotics needed. So to summarize, a substantial naturally occurring genetic variation in robustness to disease exists. So we see evidence from the literature, but also from our own internal research trials. I didn't go through all of the different experiments here, just wanted to give you kind of a taste for what that looks like. But we've done a number of different projects since looking at validating this approach of selecting for natural variation and robustness in different genetic lines, using different modes of challenge. So an experimental approach versus a natural exposure, and then also different pathogens. So different isolates of PRRS, but also different suite of co-challenges. And then demonstrated this in a proof of concept study. So that one that I just shared. So genetic improvement in robustness to disease can be facilitated using traditional breeding strategies, which we know is an effective, natural, and a sustainable approach for making genetic improvement. So I also wanted to talk a little bit here about a major change that we are starting to see and will continue to see in the swine industry in North America in coming years, which is changes in housing. So already there have been a lot of discussions about some concern, especially from producers, about how this movement towards more of group housing might impact production. And it's relevant that they're concerned, of course, because we know that changes in the environment can have an impact on animal health. Right, so flooring, for instance, is a big one. Right, if flooring is not optimal in an animal's environment per se, that can have consequences for leg health. Now, likewise, this concept of moving from individual housing to group housing can result in changes in animal behavior, right? An animal may behave differently when it's on its own versus now having to learn how to adapt to this type of group rearing environment. And there's also this connection between the two. So, for example, animals that are not feeling well may have changes in behavior, and vice versa. There's definitely this connection between those two. So one of our focus areas in response to this is aiming to breed for social sows. So we have projects looking at how to identify and breed sows that are going to perform and behave well in group settings. So classical animal models only account for the direct genetic effect. Right? So what impact does an individual's genes have on its own performance? But this type of modeling, of course, is not sufficient for traits that are influenced by group dynamics. So there we really need to investigate the potential impact that an individual's genetics has potentially on the phenotypes of its pen mates or its group mates. So we have a collaborative project with Juan Steibel and his PhD student, Andrea Nunez at Iowa State, where the objective of this work is to look at using social interaction models to estimate the effect of an individual's genetic background on its own performance versus that of its group mates for reproductive traits. So this is using a data set for group cows, gilts, and sows, and then looking at the potential impact of being reared in this group environment on these different reproductive parameters. Another aspect of sow behavior that we're focused on is mothering ability or behavior towards her young. And this is actually a trait that we've already been selecting for a number of years. And the way that we define mothering ability is the maternal effect on piglet survival. And this is a figure here from a study where we looked at the relationship between breeding value for mothering ability with her piglet's ability to get up and start consuming colostrum after birth. So what we see, you can see the three different lines here where the dark purple line are those that had the most favorable breeding value for mothering ability. And then this light purple line would be the animals with the worst breeding value for mothering ability. So piglets born to sows that had high genetic merit or high breeding value for mothering ability actually stood up, were at the udder and consuming colostrum faster than piglets born to low breeding value sows. And these are some details from our recent project of a PhD student at Wageningen University who we are partnering with, where she's looking at also the impact or the relationship of mothering ability under different housing conditions. So we had two different treatment groups of sows that were temporarily crated and those in a free farrowing system. Those in the temporarily crated treatment group were crated from 2 days prior to their due date through 5 days post-farrowing. And those in the free farrowing group were housed in these free farrowing pens throughout the duration of the study. We also estimated breeding values for mothering ability for all of the sows in both of the groups, which were just available from routine breeding value estimation. So the student, Kiara, she recorded piglet mortality reasons for every litter for pigs from 0 to 5 days post-farrowing. So she recorded overall number of piglet deaths per litter, but also specified those that were deaths due to stillborn, non-viability, crushed, and unknown. And the idea here was to estimate differences in piglet mortality reasons between those that had high versus low breeding values for mothering ability, reared in the crated versus free farrowing system. So what she showed is that when we look to this association of mothering ability with piglet losses, she saw that results from the study showed that a higher or a better, more favorable breeding value of the sow for mothering ability was associated with significantly lower overall piglet mortality. So whether that sow was in the crated environment or in the free farrowing environment, if she had a better breeding value for mothering ability, that was associated with lower piglet mortality. Most of the piglet deaths in this study were related to crushing, just given that half the sows were in that free farrowing treatment. So also saw that a higher or a better breeding value for mothering ability was associated with lower mortality due to crushing, but also with stillbirth. She did not see a significant effect on mortality due to non-viability or unknown causes. And I think that makes sense, just given that these two reasons would be seemingly less related to the behavior of the sow. So to summarize, breeding values derived under conventional conditions also work in a free farrowing system. That was a really encouraging finding for us, because all of the data that we've been collecting to date to drive breeding value estimation for mothering ability is collected in a crated system, but we see that they're also effective when sows are housed in this free farrowing environment, which is encouraging for situations where sows have to be reared in that type of environment. So next steps involve data collection at our new nucleus farm in Canada. So we have a new facility there that was populated in April of 2023. It's a 1,600 head nucleus farm for one of our maternal lines, and quite unique in the sense that it's 100% group gestation and also 100% free farrowing. So one of the reasons that this farm was designed to be a free farrowing farm is because it enables us to observe differences in behavior that we wouldn't normally see in a conventional crated system. So once you take the crates away, you're able to see these differences in maternal behavior, but also piglet behavior that we can't observe otherwise. We also have cameras that are mounted above individual farrowing pens, which are performing continuous data collection on both sows and piglets. So here's a little photo of what one of these free farrowing pens look like at the farm. You can see the sow has quite a bit of space here to move around. But also attached to this is what we call the nanny. So there's a little door here the piglets can move between the pen and the nanny as they wish. And there's also heat lamp and heated flooring here for the piglets. So there's quite a lot of data that can be extracted from this camera footage, and some of the steps that have already been taken include being able to identify individual sows and then track those sows over time, and then more recently identifying the piglets. So that's actually been even more difficult than identifying the sow, because a little bit harder to find the distinguishing features between the individual piglets. So apart from identifying the sow, also able to define this contour around the sow and then classify the position or the posture of that sow. So the researchers in our group that are working on this are now able to identify some of the different postures that you see here that the sow is performing. And then this is really a first step at using this information to identify the amount of time that the sow is spending in these different postures as a way of looking towards developing novel behavior traits to select for desired maternal behavior, and going forward, also looking at doing the same for piglet behavior. So to summarize this part, exhibiting positive social behavior towards pen mates as well as progeny is critical for success in alternative housing systems. Both of these types of behaviors are heritable. Selection for better mothers using data collected in a crated system, which is our current approach, can also be used to improve mothering ability in a free-farrowing system. And some overall conclusions for the things that I've showed here today. Now, health and behavior traits can be improved via genetic selection. We see evidence of substantial naturally occurring genetic variation in those traits. We perform selection for enhanced robustness under conventional health conditions, but also specifically under disease challenge conditions and for improved behavior. And I think this is the type of holistic approach that we really need to think about now and also going forward for improving pig health and welfare for profitable and sustainable pig production. Lots of people involved in these projects, which I want to recognize and thank. You can see that there's lots of different areas and things that have that they've contributed to over the years. So with that, I would thank you very much for your attention, and I'd be happy to take any questions that you might have. Thank you.
Thank you so much, Dr. Dunkelberger, for such a great presentation. I'll open it now to any questions in the room. I'll start with Bruno.
Hello, Dr. Jenelle. First of all, great talk. Thank you. I would like to ask you a question about the disease and robustness and I guess some anatomy problems as you have showed for us. Have you checked for any correlation between this kind of disease and inbreeding level? And how do you manage the inbreeding in your group of animals, as the characteristic of the breeding program is to use the pyramid scheme? So is there any correlation between susceptibility to disease, articulation disease problems, or even robustness, fertility disease? Because in cattle, we see that, for example, inbreed has a lot of negative effects in fertility traits, for example, and even some malformations. So I would like to listen about this.
Okay, yeah, thank you very much for the question. So maybe just to make sure I understand, the question is if there could be a potential negative impact of inbreeding on susceptibility to robustness to disease.
Yes.
Yeah, sure. Yeah, that's a great question. We haven't investigated that question specifically. I think that's a good suggestion. I think one of the questions or the comments that we've been investigating maybe more so has been, this thought that there might be a negative impact of selection for heavy emphasis on production traits on susceptibility to disease. So that's kind of where we've focused most of our efforts on this to this point, is trying to understand that potential relationship, because I think that discussion has been going on probably for a number of years as well, right? This thought that breeding for are putting a lot of selection pressure on reproductive performance, on growth, on feed efficiency, has negative consequences for robustness to disease, because those animals are simply compromised in terms of the number of resources they then have to dedicate towards mounting and sustaining an immune response. But those things, I think, could probably all be connected, because Lyme breeding, of course, is a form of inbreeding. So I think that, your point is definitely a relevant one for consideration. But we haven't investigated, or at least I haven't, from from the research side, maybe the genetic nucleus group more so investigated that specifically. Thank you.
Thank you.
Colin, you can go ahead. Colin?
Can you hear me now?
Yes. Hi.
Okay, perfect. Yeah, to follow Bruno, thank you for the great talk. A ton of work gone into all of that. My question, I have a few questions, but I'll start with one, and then if no one else has any questions, I can ask a couple more. But specifically around the robustness study that you did with respect to, I believe it was PRRS, where you had high robustness and a low robustness group, and you compared them and you found that you could have higher genetic variance, or you had a higher heritability. I was just wondering specifically, how did you house or group those animals? Did you group them specifically the low together and the high together, or do you know what the structure was for that?
Yeah, good question. And maybe the question behind your question might have something to do with the transmission or shed and spread of disease.
The social effects, because if they're all the low ones are together, then they're going to get sick and there's going to be more infection in the environment increasing. And you have the knock-on effect. Yeah,
insightful question. So the way that these animals were housed is that we grouped them on, well, let's see. Okay. Did we group them on a pen basis? I think we grouped them on a pen basis. So we had one barn where all the animals were in the study, and then every other pen we had alternating between the high and the low group. So
they weren't mixed between.
They weren't mixed between, but everybody had nose-to-nose contact.
Okay.
Yep. So within a pen, you would have, I think it was 25 to 27 pigs per pen, but then alternating side by side. So the low pigs and the high pigs all did have nose-to-nose contact with each other.
Okay. And just out of interest, did you look to fit a social effect, given that you've done it with the other traits? I was wondering for disease specifically if you've looked at that.
We haven't, but I would love to. I'm fascinated by this concept because I'm quite convinced that there's also a genetic component to disease transmission dynamics, but very difficult to come up with the proper structure to really evaluate that, right? Because I think that what we would need to do, let's just use this example of having these two extreme groups, for instance, is I think we would need to block the setup of those animals in different rooms where we would have, let's say, within one room, the shedders would be the low robust group, and then have high recipients, in another group low and low, and the other one high and high, something like that, where we could have confidence in terms of which animals would be potentially responsible for shedding and then who the recipients would be. I think that's how we would need to design that to have confidence around it. Because using the experimental model that I just described, we don't, right? With all the animals, even though they were grouped within a pen in the group, because they had that nose-to-nose contact, we don't have confidence in terms of which animal really shed to a given individual.
Yeah, for sure.
Yeah. If you have suggestions or
Yeah.
insight to there,
So
I'm
I
interested.
did, like, part of my PhD, I did it for calf diseases by fitting.
Okay.
And I worked with, I know you work with Vahaning as well, so I was working with Peter Bima over there
Yes. Yeah.
on
Yeah.
it specifically. So he does all these indirect genetic effects and stuff. And
Yes.
yes, it was quite complicated. And we basically we guessed which animals were actually exposed to each other
Okay.
at a given point in time, and then estimated respective effects for both susceptibility and infectivity too. Because the only issue I see with your point there is, though an animal might appear healthy, it might still be shedding. So the low robustness ones don't technically mean that they're the least susceptible, or the high shedding ones doesn't just necessarily mean that they're highly susceptible. They could just be very robust and still shed. So it is complicated.
Right.
To me, it would just be, once you know what the group of animals is, I think that would be sufficient, but I hadn't even thought of some of the concerns that you had regarding that. I would have just thought the perfect would be mix them together, 10 and 10, 10 high, 10 low, across the different groups, and just see which ones get sick and when did they get sick. And once we did it over the first 70 days of life, where each 10-day period was a different trait, essentially, or a different phenotype. And then across, at the end of 10 days, basically, which ones got sick. After 20 days, which ones got sick. They shed. The earlier they get sick, they're going to have a more susceptible phenotype and likely a higher infectivity because they have more time to infect others and so on. Well, we can talk about that after if you do have any questions about it.
Yeah, I'm very interested in that. Yeah, and I've had some communication with Peter on a number of projects, but also Dries Holst.
Yeah, Dries Holst. Yeah, I worked with him as well.
You did? Okay. Okay,
and
great.
Mark de Jong is the epidemiologist that they typically work with. Yeah.
Okay, well, nice to meet you. Yeah, it would be nice to have some more discussion on this topic. Yeah, thank you.
So we are over time, but if you do have time for a few more questions, just can keep going, but it's up to you. Okay. Okay. Maria, do you want to ask your question?
Can you hear me?
Yes. Yep.
Hey, Jenelle, I'm Maria from the university, and my advisor just shared the link of her presentation this morning, and I'm so glad that he did because I really enjoy it. Thank you so much. Well, my question is regarding some economic values. Have you analyzed the economic values on the welfare, health, and the resilience traits? Because although we as animal scientists and veterinarians, we care more about welfare. I know we also live in a capitalist world, so topics as a company, how do you deal with this? Like, how are they calculated? Maybe adding these traits can impact negatively in productivity, or I don't know, do you think about the market? How I want to know more about it.
Oh, that's a tough question, Maria. Yeah, thank you. Thankfully, I could say I'm part of the research group. So when it comes to deriving economic values, we have a whole team of people that are that I can call on for that part. But I'm part of the discussion in terms of how we go about that. And actually, this has been kind of a difficult conversation around this concept of bringing this breeding value for robustness to disease into light, because as I described it, it is meant to be comprehensive of an individual's response to whatever type of disease challenge that it might encounter. But there are some markets in which we do business where this type of intense exposure to disease is not really relevant. Right. So for instance, Norway is a major market for us because it's one of our home markets, but they're one of the cleanest countries globally when it comes to disease. So breeding for enhanced overall robustness to disease is not going to have the economic relevance for them that it does for the US and that it does for China. But at the end of the day, we have one selection index to rule them all, Right? We don't have sub-indices for selecting animals for these different markets. So there is a balance that we have to come to in terms of how much weight do we put on this trait, recognizing that it is going to be more economically relevant for some of our markets than others. Another important factor that we also have to take into consideration is the incidence. Right, so even if we look to a market like, the US, Mexico, China, where outbreaks, there is a higher likelihood of outbreaks, it's still not 100% of the time. So then we also have to think about putting an estimate in there in terms of, okay, surveying what's the kind of incidence rate of disease that we might encounter, and then also taking that into consideration. So we actually haven't landed at a conclusion on this yet. This discussion is still ongoing, which I guess maybe to also your point kind of indicates the difficulty of coming to this, because everybody also comes with a different opinion in terms of how we should end up making a conclusion on it. But yeah, those being some of the different factors involved in the discussion.
Thank you, Jenelle.
Thank you.
Thank you. Harry, do you want to ask your question?
Yeah, I'll be quick. Thanks. Hi, Jenelle. It's Carrie. I'm from Genesis. And fantastic presentation. Really interesting. I had one question and I might have missed it. With
Sure.
the work that you guys have done on sort of that free farrowing, have you seen any results at this point in terms of either a genetic or a phenotypic change on improved mothering ability in the free farrowing housing since you've started to work with that?
I think it's still too early to see that. How should I
I
say
know
this?
it's not been long, right? So
Right. Yeah, it's pretty early on that we've started collecting data in the free farrowing environment. So I think I would preface it from that perspective first, that we probably don't have enough data collected in a free farrowing environment to really, let's say, make any conclusions about the impact of selecting for mothering ability in that kind of time frame. However, we can see from the study that I showed where we had those two different treatment groups of the conventionally housed sows versus the free farrowing sows, that if you collect data for mothering ability in a conventional system, it's also predictive of mothering ability in the free farrowing environment. So I think that's the main thing, is that, even historical data that you've collected that can be used to calculate genetic merit for mothering ability, it's not obsolete when you put those sows in a free farrowing environment. You can still identify better mothers in a free farrowing environment using those data. So I think that's encouraging, from that standpoint, that we can still use that data, that historical data, to select in that direction.
yeah, fantastic. Thank you.
Thank you so much. If there aren't any other short questions, I think we'll stop here. But it was a great discussion. Thank you again, Dr. Dunkelberger, for presenting with us today. The recording for this will be posted on to YouTube, and we can send you those details later. I did also want to make an announcement on the upcoming schedule for CGIL Seminar. Our next presentation won't be until Friday, February 28th, but we are looking forward to seeing everybody then. And with that, I will thank you all for coming. It was a great discussion. Thank you.
Thanks so much for the invite. Really enjoyed it. Thank you. Bye.
You're laughing on my notes?
Yeah, no, for